Good morning, everyone. Today I'm going to present my seminar on nicotinic hypothesis of schizophrenia. So first of all, we'll go through what are the all neurotransmitters involved in the pathophysiology of schizophrenia. First is the dopamine hypothesis, which says that if there is a hyperactivity of dopamine D2 receptor transmission in the subcortical limbic region, that is in the mesolimbic pathway, that will lead to positive symptoms. And if there is hypoactivity of dopamine D1 receptor in the prefrontal cortex, that is the mesocortical pathway, that will lead to the negative symptoms. Then the second is the glutamate hypothesis. The glutamate hypothesis, this is the hypothesis which says that the dopamine hyperactivity is a consequence of glutamate dysregulation in the prefrontal cortex. So here in this diagram, as we can see, this is a diagram of a prefrontal cortex. There are pyramidal cells. There is a GABA interneuron. Okay, so the pyramidal cells, they secrete glutamate. Glutamate is a excitatory neurotransmitter. So when the glutamate is released in the surrounding, the GABA interneuron, which has NMDA receptors on them, sense the presence of glutamate and then send a negative feedback to these pyramidal cells to stop the secretion of the glutamate so as to prevent glutamate toxicity. So in schizophrenia, the hypothesis is that there is dysfunction of these NMDA receptors, because of which the negative feedback is not there, because of which the glutamate is released in excess quantities, leading to excessive dopamine release in the ventral tegmental area. Third neurotransmitter is GABA. The GABA is synthesized from glutamate by glutamic acid decarboxylase enzyme. It has two isoforms, GAD65 and GAD67. In schizophrenia, it is a finding that there is reduction in GAD67 mRNA levels in the prefrontal cortex. And because of which there is loss of inhibitory GABAergic neurons, leading to hyperactivity of dopaminergic neurons. Then is acetylcholine. Acetylcholine is cholinergic neurotransmission, is known to be integral for cognition, memory functions, and which is disrupted in schizophrenia. So decrease in level of nicotinic and muscarinic receptors in hippocampus, frontal cortex, thalamus, and striatum is seen in schizophrenia. There are abnormalities in alpha 7 and alpha 4 beta 2 nicotinic acetylcholine receptors, and also there is decrease in the M1, that is the nicotinic muscarinic receptor in the DLPFC and hippocampus, as seen in the postmortem findings. Then there is a serotonin hyperfunction hypothesis. This hyperfunction hypothesis is mainly described in elderly patients with dementia, in which they have described that like in elderly patient, due to deposition of plaques, there is loss of GABAergic inhibitory neuron. So GABAergic inhibitory neuron on these glutamate cells, there are serotonergic receptors which give which give positive feedback to to these glutamate cells, and GABAergic interneurons, they give negative feedback. And because of the disturbance of because of the loss of this negative feedback, there is excessive serotonin, thus causing serotonin hyperfunction. Now, the cholinergic system in schizophrenia. So the cholinergic system, it consists of two receptors, nicotinic acetylcholinesterase receptors and muscarinic acetylcholinesterase receptors. Both these receptors are found in have been found to modulate brain circuitry dysregulated in schizophrenia. So the nicotinic acetylcholinesterase receptors, these are ligand-gated ion channels. These are pentameric structures made up of these are ligand-gated ion channels, and they're of two types: muscle receptors and neuronal receptors. The muscle receptors, they are made up of alpha, beta, gamma, delta, and epsilon, 5 subunits, and the neuronal receptors, they are made up of alpha and beta subunits. So in alpha and beta subunits, they are mostly made up of alpha 2 to alpha 10 and beta 2 to beta 4 subunits. subunits. So various combination of these subunits make distinct receptors that vary widely in their pharmacological and biophysical properties. And the most widely distributed receptors in the brain are alpha 7 and alpha 4 beta 2 receptors. So whenever the nicotine comes and attached to these receptor, the ion channels of these receptors get activated, and this result in the movement of sodium and cations across the cell membrane, triggering the voltage gated calcium channels to open and ultimately release of the neurotransmitter. So nicotine basically modulates release of all the neurotransmitters, including dopamine, glutamate, non-adrenaline, serotonin, acetylcholine, and GABA. Then comes the muscarinic acetylcholinesterase receptors. They are mostly these receptors. They are M1 to M4 receptors. The M1 and M4 receptors, they are implicated in psychosis. Polymorphism of these receptor subtypes has also been seen in schizophrenia. So M1, M3, and M5 receptors, they are excitatory receptors, and M2 and M4 receptors, they are inhibitory receptors. So M1 receptor, which is a excitatory receptor, is present post-synaptically, and it is present the highest density in cortical regions and in hippocampus. And the M4, which is a presynaptic autoreceptor and hence the inhibitory receptor, is highest in the striatum, that is 20% of the total muscarinic receptors. These all receptors also, they modulate the signaling, the release of dopamine, GABA, and glutamate neurotransmitters. Now, why do people with schizophrenia smoke? The prevalence of prevalence of smoking in schizophrenia is around 70 to 80%. So the first thing is cigarette smoking is a consequence of psychotic disorder itself. And this could arise because of boredom, apathy, and reduced motivation as a part of deficit syndrome. As a self-medication, nicotine regulates dysfunctional mesolimbic dopamine system and increase dopamine release in the prefrontal cortex and alleviate positive symptoms. Patients with schizophrenia, they may smoke heavily as a result of antipsychotic medications which produce marked dopamine receptor blockade. So a very high level of smoking is necessary to overcome this blockade and produce reward. And to improve cognitive deficits in individuals with psychosis, including work, memory, attention, and gating deficits. So this is a theory of reverse causation, that is the smoking is a consequence of psychotic disorder itself. Now, what are the various gating deficits or markers of vulnerability or endophenotypes in schizophrenia? First is P50. So what happens whenever there is a stimulus, whenever we give a stimulus, so our cell membranes, they go into hyperpolarization and they become refractory. So whenever a second stimulus will be given within a few seconds, so the result will not be that. The result will not be similar, it will be diminished. So normally the response to the presentation of a paired auditory, in P50 we give a paired auditory stimulus, and we'll see that there is 80% diminution of the second wave related to the first due to activation of inhibitory neural circuitry in the first auditory stimulus. So P50 is an electroencephalographic finding of cortical inhibition that occur when the second stimulus is presented after 500 milliseconds. So in schizophrenia, schizophrenia is associated with diminished inhibition of the second stimulus, that is diminished P50 inhibition. This deficit is genetically linked to polymorphism found in the promoter region of CHRNA7 coding gene for alpha-7 nicotinic subunit, as well as decreased functioning of alpha-7 nicotinic acetylcholinesterase choline receptors. Nicotine, when it was seen that when nicotine was given, this deficit was cured, and nicotine remediated the deficits of P50 suppression. Then the second vulnerability marker or endophenotype is pre-pulse inhibition. It is an electromyographic finding. So when PPI is an electromyographic measure of eye blink response to a startled auditory tone. If a pre-pulse tone occurs before the main auditory stimulus, if we give a small stimulus before the main auditory stimulus, this eye blinking response gets diminished. So in schizophrenia patient, patient have deficit in this inhibitory response, and this deficit is associated with CHRNA3 polymorphism relating to nicotinic acetylcholine receptor dysfunction that is characteristic of schizophrenia. And again, nicotine improves this deficit in smokers with or without schizophrenia. Third is mismatch negativity. Mismatch negativity is that when a series of standard stimulus are given and a deviant stimulus is presented in between, or an oddball stimulus that we say, it result in evoked response that can be recorded on EEG. So the negative component of the this mismatch negativity, this component is obtained by subtracting the event related response to a standard stimuli from to a event related response to a deviant stimuli, and it usually occurs after 100 to 250 milliseconds. And the significance of this MMN is that it indicates the patient has the ability to automatically distinguisch and induce attention to two physical stimuli known as pre-attentive process. Again, this deficit is seen in schizophrenia, and nicotine enhances the duration of MMN amplitude and facilitate improvement in this deficit. Last is smooth eye pursuit movements. So this is basically a test. Smooth eye pursuit typically measured by human in human by in laboratory by presenting a stationary object with a small visual target such as a dot on a computer screen or a light emitting diode, moving slowly in the horizontal plane and requesting the subject to follow the target with their eyes while keeping their head still. So the key performance measure is like the patient will follow the person will follow the direction of our target. But and then a anti-second task is given. In this anti-second task, a new target is given to the patient, and he is asked to move the eye opposite to that task. So in schizophrenia patient, they show impaired smooth pursuit eye movement and generate a high percentage of error on anti-second task compared to a healthy individual, and this deficit again get corrected by nicotine administration. Now, the next theory is that genetic or environmental factors might both lead to nicotine addiction and schizophrenia. So this says that genes for psychosis overlap with that of cigarette smoking. So schizophrenia working group identified 108 genome-wide significant loci associated with increased risk of schizophrenia. One of this was CHRNA5A3B4, which was on chromosome 15q25, which code for alpha 5, alpha 3, beta 4 nicotinic receptor subunit. This gene and also this gene has strongest genetic contribution for nicotine dependence. Another was genetic polymorphism in gene CHRNA7 coding for alpha 7 subunit of nicotinic receptor, increase the risk for both schizophrenia and nicotine dependence. The other genes that are common in schizophrenia and smoking is BDNF gene polymorphism, dopamine beta hydroxylase gene, mu opioid receptor gene OPRM1, and dopamine 2 receptor gene DRD2. So this was a theory of shared liability. Now the last is smoking might be the etiological factor for schizophrenia. So 4 recent meta-analysis conducted each concluded that smoking increased the risk of psychotic disorder. So this was the first meta-analysis by Miles et al. That examined the prevalence and course of tobacco use during early psychosis in a meta-analysis of 58 studies. And they found that individuals with first episode psychosis tended to have been smokers before the onset of illness, to be heavy smokers at the time of presentation of treatment, and were much more likely to smoke than age-matched healthy controls. This was the second meta-analysis by Gorillo et al. They examined 61 cross-sectional case-control prospective studies revealing that smoking increased the risk of psychosis. Then Hunter et al. In 2017, they analyzed 5 prospective studies and concluded that smoking had a significantly increased risk of developing schizophrenia. And lastly, this study by Scott et al. Meta-analysis done in 2020 of 7 cohort studies and 1 cross-sectional studies, and they identified smoking as a significant risk factor for schizophrenia. Now, if cigarette smoking is causally related to psychosis, how this fit in our current understanding of neurobiology of schizophrenia? Now, the neurodevelopmental model of schizophrenia, what it says is that a genetic predisposition along with environmental, social, psychological factors will lead to neurodevelopmental abnormalities and target features that will lead to brain dysfunction, improper balance of chemicals, and ultimately schizophrenia. So, in continuation to that, prenatal exposure to smoke increase the risk of schizophrenia by 29%. The studies have shown that fetal exposure to nicotine via maternal smoking during gestation was related to increased odds ratio of schizophrenia in offspring. It is associated with structural brain changes like cortical thinning, decreased corpus callosum volume, and these brain abnormalities are also seen in schizophrenia. There is abnormal development of cerebral inhibitory neurons like GABA as found in schizophrenia. And it is also associated with epigenetic methylation effect, leading to downregulation of BDNF, which is related to schizophrenia. Now, the second is smoking. The smoking can cause activation of the resident cells and recruitment of inflammatory cells into the lungs, which lead to release of pro-inflammatory cytokines, chemotactic factors, oxygen radical proteases, which alter the functions of immune cells. It is highly associated with smoking is highly associated with the release and inhibition of pro-inflammatory and anti-inflammatory mediators. Uh, and inflammation process underlie a number of physical health consequences of smoking. Inflammatory cytokines also modulate dopaminergic and glutaminergic neurotransmissions directly. So a genetically susceptible individual to schizophrenia plus late life stress or immune activation will act on the prime microglia or immune cell, leading to overactivation and aberrant synaptic pruning. So the microglia activation secondary to two hits may lead to excessive pruning of neurons by microglia in the stress-sensitive areas such as prefrontal cortex and hippocampus, and which will lead to loss of synapse during pruning by structural changes in which will lead to structural changes in the brain associated with schizophrenia and development of negative and cognitive. Again, inflammation, it can activate the HPA axis leading to release of cortisols. Also, cigarette smoking alters the metabolism of cortisol. So persistent increased cortisol cause neurotoxicity and death of neurons in hippocampus, where highest glucocorticoid receptors are present, leading to loss in hippocampal volume. Similar changes are seen in schizophrenia also. The constituent of cigarette smoking have significant effect on the endocrine system by altering production and metabolism of estradiol, which contribute to sex differences in schizophrenia. Now, a work in animal model shows that nicotine exposure increases D2 high affinity receptors, thus supporting the role of nicotine as one of the schizophrenia risk factors. Nicotine also alters the release of almost all major neurotransmitters implicated in the pathogenesis of psychosis. Now, the novel treatments beyond the dopaminergic receptors, what are all the novel treatments? So first is xanomeline and trospium chloride. So xanomeline, as we had studied that the muscarinic M1 and M4 receptors were included in smoking. So preferentially M1 and M4 muscarinic receptor agonists. This is a preferential M1, M4 muscarinic receptor agonist. And trospium chloride, it is a peripherally active anticholinergic drug, and it does not cross blood-brain barrier. So it was added so as to avoid the side effect, gastrointestinal side effect of xanomeline. So combination of xanomeline, trospium chloride designed to reduce the frequency and severity of peripheral procholinergic side effect. And this drug is currently in phase 3 trial. The next drug is emraclidine, which is a positive allosteric modulator that selectively act on M4 muscarinic receptor, and it is in the phase 2 clinical trial. Now, galantamine is a positive allosteric modulator, which positive allosteric modulator affect receptor function by interacting at secondary or allosteric binding sites, where they facilitate the effect of endogenous neurotransmitter. So galantamine is an anticholinesterase inhibitor, and also it is an allosteric modulator to alpha 4 beta 2 and alpha 7 nicotinic receptors. It is indicated for the treatment of mild to moderate dementia and Alzheimer's disease. And the most common adverse effects are nausea, vomiting, diarrhea, dizziness, headache, and decreased appetite. One more is TC1734, which is a selective alpha 7 receptor agonist. It is in phase 2B trial for the treatment of cognitive deficits in schizophrenia. Pimavanserin, it is an inverse agonist of 5-HT2A receptors. It has high affinity for 5-HT2A receptors with lower affinity for 5-HT2C receptors and almost no affinity for 2B dopaminergic, muscarinic, and opioid receptors. It is the first FDA-approved drug for the treatment of psychotic symptoms in patients with Parkinson's disease. Then is sodium benzoate. It is an NMDA receptor hypofunction. So as we all know that there is NMDA receptor hypofunction in schizophrenia. So D-amino acid oxidase degrades the D-serine, which acts as a co-agonist of NMDA receptor. So in NMDA receptor, there are two co-agonists, D-serine and glycine. So D-amino acid oxidase degrades this D-serine. So sodium benzoate is the inhibitor of this D-amino acid oxidase, useful in treating schizophrenia by preventing peripheral breakdown of DC. So there are two ongoing phase 2 and phase 3 clinical trials on safety and efficacy as an add-on treatment for schizophrenia, one in adolescents and one in adults for this drug. Last is clepertin. It is again we know that there is NMDA receptor hypofunctioning in schizophrenia. So glycine is a co-agonist for NMDA receptor. So clepertin, clepertin, it is a glycine transporter type 1 inhibitor, and it is in phase 2 randomized double-blind placebo-controlled trial. The phase 2 study is evaluating clepertin added to current antipsychotic therapy and computer-based training for cognitive symptoms of schizophrenia, and phase 3 studies of add-on therapy with clepertin are underway. These are my references.
So floor is open. Questions, comments, or clarifications? Declaration.
Nicotine helps in cognitive deficit, but also there are studies which
No, interesting question, because if someone is reporting